I want to talk a little bit now. We're going to move on to clinical development. Certainly encourage our audience to continue to submit questions. We will be getting to them. David, I suppose I'll start with you here. What is your company, what is Forge's perspective on this? The overall clinical development strategy for AAV-based gene therapies. What are you hearing from your partners? What in your former roles, what would you say is kind of the best strategy from a clinical development standpoint?
Make sure I'm understanding the question. Sorry. Sorry, repeat the question. I apologize.
Yeah, sure. The overall clinical development strategy for AAV-based gene therapies, best practices, what does it look like?
Yeah, so make sure I'm answering your question correctly. But as far as the strategy for your overall clinical development, I think what we've seen is having really good models, right? Your preclinical models, then to know what your dosing should be to get that distribution, and then leveraging that information, and really having, ideally, I think the big questions are really around biomarkers and how those can be leveraged to understand more quickly what the impact of the therapy is having, especially in some of the neurological diseases where the readout can be years. Having those biomarkers and having a really good understanding of your vector is pretty critical. And where we see, I think, people struggle a bit is really around the analytics and having the analytical assays early, and really investing in that, because really understanding your vector and some of the nuances around the vector, whether it's a knockdown approach or it's a gene replacement approach or a gene editing approach. I think oftentimes it's analytical, both from clinical samples as well as from release of the vector. Understanding how investing more in that earlier in development, and instead of waiting later on, critically, like we see potency assays being pushed, really true functional potency assays being pushed till later in development. But I think that's something that can be really critical to understand critical quality attributes and how they impact potency of the vector. So I hope that answers your question.
It does, yes. Yeah. Thank you. And then I think, what we see on Cell and Gene too, from our subject matter experts who we work with, they forever are saying, timing is everything, the sooner the better. Nina, I have a question for you. How does REGENXBIO prioritize which AAV programs to advance into clinical trials?
Yeah, so I think first and always is understanding patient needs. Case by case, right? We want to understand the specific unmet need here. What is important to patients and their families? What is meaningful to patients and their families as we think about the different programs? And then on the more technical part of this question, I think we mentioned a lot of this already, the technical feasibility, right? So the capsids, do we have the ability to target the tissues of the cells that we want? What would be the route of delivery? As David mentioned, animal models and ways to build a robust preclinical package to be able to move forward. And then, of course, looking at the competitive landscape, right? So what does that look like in the cell and gene therapy space, especially if it's for a rare disease? We think a lot about time to clinical proof of concept, and this is related to biomarkers and potential surrogate endpoints or intermediate clinical endpoints, as David pointed out as well. And then, of course, regulatory flexibility. Like, are there ways to think about being able to be more efficient as we move certain programs forward, and then manufacturing scalability? I think that's something to think about so that we can really have a more holistic end-to-end sort of look at the different programs and help us prioritize.